So I'm Richard Scheines. I'm at Carnegie Mellon University. This is my colleague Greg Cooper at the University of Pittsburgh. And we're going to give you a workshop on computational causal discovery and applying it to biomedical research. So we're going to bounce back and forth between a little bit of theory, understanding what basically is driving this entire perspective, and then using the Tetrad program, which is giving you a hands-on experience of building these models, searching for these structures, et cetera, et cetera. And the data that we're going to use is available both on the conference box site, but also on this separate site we set up so that you don't have to have a password, and it's much easier to get.
And it's on the first.
Yeah, it's on that first newsprint on the left there, in case, because I'm not going to stay in the slide for very long. And the program that we're going to actually use, we sent email for you to download. And let me get a show of hands. How many people have successfully downloaded this program and can actually use it? So we're going to spend a few minutes motivating the topic, and then we're going to cover the basics of causal graphical models, and then we're going to transition to what technology we've developed and others have developed to search for causal structures. And then we're going to go through, we'll go through along the way a bunch of examples to give you a more concrete sense of what's happening. Okay, so in motivating why we want to actually represent and search for causal structure, there's a number of things we do in science and in everyday life that require causal knowledge. And the first one is counterfactual reasoning, right? So if I'm late to work and I say, well, if I had set an alarm, I would not have been late to work, I'm sort of depending on a structure that looks something like this, which is I set the alarm, that wakes me up at 7 and I get to work by 8. But if I incorrectly say I didn't finish that marathon, and I would have finished the marathon if I had just dyed my hair black, right? That's a false counterfactual, and you can see it's false because what's really going on is that, right, being old like I am causes me to have low endurance, and it also causes me to have gray hair. So the fact that gray hair predicts my lack of endurance doesn't mean I can paint my hair black and actually increase my endurance. And that's because of the causal structure that I'm showing you there. So those simple cases really say quite a lot about the whole thing. If we want to do any policy reasoning in medicine or other areas, we have to have causal knowledge. I know that if I raise taxes on beer, I will lower underage consumption. And I know that if I want to lower infant mortality, that'll actually lower the birth rate. But if I make people wear shorts in Pittsburgh in January, I will not make it warmer, right? And those are just another simple set of cases about policy. And it turns out that explanation in science also requires causal knowledge. So in this classic example from the philosophy of science, we're trying to explain how long the shadow is. And if we know the position of the sun and the height of the flagpole, then we can deduce the length of the shadow, and we can explain why the shadow is 10 feet, 8 feet, or whatever it is. But if we're actually trying to explain why the flagpole is the height it is, and I see how long the shadow is and where the position of the sun is, I can deduce from geometry the height of the flagpole, but I will not have explained it. I will just have deduced it. And so explanation requires causal knowledge as well. Observational prediction, by that I mean observing something and predicting from it, does not require causal knowledge. Right? So if I watch people walking around and I'm saying everybody's carrying umbrellas at noon, right, I can still predict that it's going to rain soon thereafter, even though making people put their umbrellas away will not have any effect on what happens. And if I'm looking among children ages 5 to 10, it turns out that their shoe size is a very good predictor of their reading level. And obviously changing their shoe size is not going to affect their reading level. So in general, I try to represent probabilistic or statistical ideas with green font and causal ideas with red, because it's helpful, I think, to distinguish the two in lots of different ways. So if I'm trying to predict from observation, that's not the same as predicting from intervention. And so this is a theme that will come up, so let me go through this a little bit. So going back to the gray hair and running the mile example, if I'm looking at what's the probability that someone could run a mile under 7 minutes, given I observe they have gray hair, that's not the same as what's the probability that they could run the mile under 7 minutes given I paint their hair gray, which is the do operator that, right, has been represented with Pearl's work and in other ways in other people's work. So this is sometimes the same, but it's not always the same, and the fact that they're different really motivates, in some sense, the whole topic. And so in general, the conditional probability of y on x and z is not always the same as the probability of y given I've done something interventionally to x and observed z. And so if I'm trying to predict causally versus statistically, I need to know more. And so in statistical prediction, if I have non-experimental data, I've done a survey or I've done a study, and I have background knowledge, I can often use those to estimate the joint distribution among the variables in the population, which I notate as p, x, y, z. And from that, if I have the joint distribution as my estimate, I can also then compute the conditional distribution of y given x and z, right, for any value of y, x, and z. But if I want to know the probability of y given I've done something to x, I need more than just the joint. I need the causal structure among the x, y's, and z's, and the joint distribution that the population has as well. So I need to get to, right, in the work we're doing, this particular thing here. How can I get from data in non-experimental or experimental situations, how can I get to causal structure? What's causing what? And that's really the task or the project that Greg and I have been working on for over 30 years now. And the good news is that there's been huge advances in this field in the last 30 years. We have a very, very sophisticated and fairly complete mathematical representation of causal systems, and from those we can derive counterfactuals, policy predictions, etc. And we have a graphical representation that's extremely useful for all kinds of other things. And from those representations, we have been able to produce fairly fast and reasonably reliable search algorithms that actually get us from background knowledge and data to something about what's causing what. And the books I've listed and pictured here are all really good references to start out. And so really we have what we think of as a modern theory of statistical causal models, which involves graphical models representing interventions and manipulation, counterfactuals, entailing from causal structure testable constraints that I can look at in data, and then discovery algorithms that we'll get to soon. So the context that we work in is very useful in situations when you don't have a lot of background knowledge. But if you do have a lot of background knowledge, often what's really done is what we call causal estimation. If I can build a model, and I'm confident in that model, and I have one parameter in the model I need to estimate, right, that's called, what we call it is causal estimation. Right? And so here's an example. What was the effect of azidovudine on survival among HIV-positive men? This is a complicated thing to estimate because of time-varying confounding, but Miguel Hernan and his colleagues figured out how to do it on the basis of several assumptions. And what they got was an estimate of the causal effect with confidence intervals bounding that estimate. The fundamental problem with this causal estimation is that all the estimation and inference you do is conditional on the model being specified correctly. So if you know the model and it's correct and you have to estimate a parameter, right, then you can get all that inference and estimation off the ground. But if you don't know that the model is correct, you have a much, much different kind of problem. And so in causal search, right, well, we don't have enough background knowledge to pin down everything except for one parameter. It's oftentimes extremely useful to have, right, a way to actually search through, you know, the billions and trillions of hypotheses that might be true and narrow to ones that are at least consistent with the data. And so here's a question that Greg is going to actually illustrate in a little more detail later, in which people were trying to figure out among the 25,000 genes in the flowering plant Arabidopsis, which of those genes were regulating flowering time. Because if they could shorten flowering time, they could grow more flowers and make a lot more money. I think this was in Holland. And so we actually worked with people in Zurich, and they took microarray measurements and came up with a very plausible set of possible candidates and then tested them, and Greg will tell you the results in a few minutes. experiments. And so the idea here is generate hypotheses and actually follow up with experiments to confirm whether you have causes or not. And the fundamental problem for this approach is that the number of causal hypotheses grows super exponentially with the number of variables. And so the search has to be extremely efficient and intelligent. Okay, and now we're going to tag team back and forth. And now Greg's going to talk about what is the basic way we fit this into a workflow that scientists engage in.
Thank you, and good afternoon. So we just wanted to basically lay out in these next few slides sort of the basic idea of what we're trying to accomplish here. Here, we typically have, in biomedical science or clinical science, we have prior knowledge, oftentimes a lot. But we've got some data also that we want to analyze. What we're going to be talking about and have been working on are methods for doing the causal analysis and producing causal graphs. We can use both observational data, which doesn't involve any experimental manipulation. An electronic medical record would be a classic example of that. We can also use experimental data like you might have with an RCT in a clinical sphere, or you may have with wet lab type of intervention experiments for biomedical research. There are algorithms that can use one of these, observational, algorithms that can use experimental. There's algorithms that can use both together. But when the purpose of these causal graphs is not to output the truth and then have that be published. It's really to help scientists develop causal hypotheses that oftentimes are true, so that it's very efficient if they follow up and test them. The main goal then, a little bit more formally, is to develop methods that suggest causal hypotheses that are novel, significant, and likely to be valid. These then would lead to experiments, and then those experiments, based on their results, would lead to additional knowledge, hopefully, additional data based on the results of the experiments, and then this cycle continues. So really in this cycle, what we're trying to do with these tools that we're going to be talking about in this workshop is improve the efficiency of this cycle by helping the scientists develop insights into what's going on causally that let them focus on the parts of the system they're interested in that are most likely to be causally true based on the output of these graphs, and then lead to experiments that are more likely to be positive. So to give an example of that, as Richard mentioned, I want to talk about some work that was done, this paper by Steckhoven, published in Bioinformatics in 2012. And they were looking at the flowering time of Arabidopsis. This is a photo of this small plant. It's sort of the E. coli of the plant world. It's been studied extensively, so there are gene libraries, knockout libraries, and overexpression libraries that are available for this plant. The observational data that they had, that they're evaluating, is 47 gene expression profiles of seedlings of the Arabidopsis. And then they also had information on a subsequent flowering time after these are planted. And in terms of data, there were Affymetric expression array measurements on a little more than 21,000 genes in this plant species. So going back to our diagram, this is the basic part of the beginning of the workflow. We've got on the left the data that's available, these microarray measurements, and then the flowering time will represent as Y. There was an algorithm that these authors had developed called C-star, and a graphical causal modeling discovery algorithm. And it produced a causal graph on the variables showing the relationship between X and Y according to its analysis of the causal relationships among those variables. What they were trying to do is find the gene expression, the genes that basically were driving the flowering time. So they selected the genes that were predicted by their the graph output by their algorithm that reliably caused the most substantial changes in flowering time. They were looking for genes that were driving flowering time according to their algorithm. Those then became candidate gene regulators of flowering time, and they followed up with some experiments I'll talk about. So they considered 25 genes in terms of the output of their algorithm that caused the most substantial changes in flowering time according to their graph analysis. 5 of those 25 genes were already known to affect flowering time, so they didn't further investigate those. But of the remaining 20, there were 13 that were not known to be regulators, and mutant seeds for each of them were available from a stock set. So on the basis of that, they performed experiments on these 13 for which they had mutant seeds for each of these genes. And a little bit of experimental detail. They used seeds for wild type and control, and then tDNA homozygous insertion mutants that were obtained from this stock center. The seeds were plated under standard conditions and grown for 10 days and transferred to soil. And then they observed flowering time that was measured in days. And the seeds that yielded 4 or more plants were considered for further analysis. They wanted to look at the mean flowering time, so they wanted to have a reasonable sample. So in terms of the results then, there were 9 of those 13 seeds that each with a single gene insertion that yielded 4 or more plants. And of those 9, there were 4 that had a statistically significantly shorter flowering time than the control wild type plants. Overall then, 4 out of the 13, or 31%, of the gene-altered seeds produced statistically significant shorter flowering time than did the wild-type plants. So it was a pretty high efficiency in terms of identifying genes that affected flowering time. So, and I wanted to mention also, they also found that of the 5 out of the 25 that were known in the literature to affect flowering time, when they did an analysis of that relative to just randomly selecting genes, that was a much higher rate, 5 out of the 25, much higher rate than was random selection relative to known flowering time genes. So they concluded that this method was finding flowering time efficiently. Now we want to switch over from motivation to talking more about the basics of causal graphical models.
Okay, so I'm going to shift gears now and sort of go into what are the basics. And this also is a topic in which we sent email about online modules that were available that will actually give you all the basics about this so you can actually use the theory a little bit more. So I'm going to break this up into a few different pieces. I'm going to talk about representing the qualitative causal structure of a system by using directed graphs. And then we're going to talk about representing manipulations and interventions in that framework, and then we're going to add probability and numbers, because that actually is required to get to a statistical model. So causal graphs are the simplest possible objects you can imagine. They just have boxes, in this case variables in general, that are connected with arrows which represent direct causes, right? So I have an edge in the system if, right, the variable that's at the giving end of the edge is a direct cause of the arrow receiving end of the edge relative to the other variables in the system. So the only thing that's a little bit subtle here is that the idea of direct causation is completely relative to a system you're looking at. There's no absolute idea here. And so if I have the years of education causing income, I think everybody would agree that's true, and that's a direct cause relative to this system. But if I add another variable that represents all the skills and all the knowledge you have at any one time, I think it's defensible to say that whatever education you receive, if it has an effect on income, it's through something you've learned, right, or something you can now do, including your social network of contacts who might get you a job. So the whole idea of directed graphs is really just that. And now I want to switch and just show you how to get into the program and build a directed graph, and then from that we can actually begin a whole set of investigations on that. Okay, so if you look at the program as a blank workbench, it has a bunch of buttons on the left, and these are objects that are going to be on the workbench and then have relationships to each other. So on the very top, there's this thing called graph. If I click it with my left button, the cursor turns into a cross. If I click that onto the workbench, it deposits an empty graph box, which looks gray. And I can do that as many times as I want. So if I double click on one of these guys, it gives me a choice, and in the simplest of things for this exercise, choose directed acyclic graph. And say OK. And then it opens up an editor, which allows you to build a causal graph by using variables that are measured, variables that are latent, and then edges between them. And so we'll leave out latent variables for now. So if I want to build a simple graph that I was showing before, I just click, 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 and then I can click on this tool and then click the left button, hold it down and drag, and let it go nearby the other guy, and arrows will be deposited. And if I want to move a variable at any time, I can click on the selection blocks there, grab this and move it, and the arrows will follow. While Brian's helping, I will demonstrate a very simple way you can rename these variables at your convenience. So if I want to do that example and click on this, I get a name box that comes up, and then I can just simply type age, hit OK, right? And now I have the example that I did before. So why don't you guys all go ahead and enter a graph like this, and then enter another graph of 4 variables completely of your choice. And I'll give you a few minutes to do that, and then we'll walk around and we'll continue. And you'll notice when the boxes are filled or populated, they turn blue to represent the fact that there's something in that box. And if you just put them on the workbench before there's something in them, they're gray to represent there is nothing in that box. Okay, so that's literally all I'm going to talk about in terms of causal graph. They're that simple. And now I want to actually model or teach you or explain to you how we model ideal interventions in this framework. So the idea of an intervention is we're going to do something in the world that's going to actually take over the natural course of affairs and change the value of some variable, or at least its probability distribution. And one of the keys, I think, to causal theories is that when we intervene on 2 variables, intervening on the effect in that pair, if there is one, has a different result than intervening on the cause. So consider this simple 2-variable system, where the temperature of the room has a causal influence on what you're wearing. If you're wearing a sweater, or if you're wearing just a shirt, I took off my jacket because the room is warm, right? If the room was going to go down 10 degrees, I put my jacket back on. So I think it's incontroversial that this is the causal relationship. And now, if I say, let's consider an intervention where for each one of you I flip a coin, and if that comes up heads, I make you put your sweater on or your jacket on, and if it comes up tails, I make you take your sweater off or take your jacket off, right? So now, what's happening when I'm doing that kind of intervention? I am eliminating the influence that room temperature had on whether you were wanting to wear something, and now I'm taking over, right? The coin flip is now determining the assignment of your clothing wearing. And so I've eliminated the edge from room temperature to sweaters on because that intervention was now taking over the value of sweaters on. Now, if I consider the same system, but now I intervene on the cause, right? So I flip a coin, and if it comes up heads, I make this room 85 degrees, and if it comes up tails, I make the room 45 degrees, right? Which I can represent by a pair of die and then arrow into room temperature. I'm going to change the value of room temperature, but I'm not going to change the relationship between room temperature and sweaters on. That causal relationship will exist, so what I'm going to change is the value of that. I'm going to leave the relationship intact, whereas in the prior setting, I changed the value of this, and I also annihilated the causal relationship because I took over. So that's the asymmetry, right, in intervention, and that makes modeling intervention actually quite easy. So we model an ideal intervention, by which I mean we're going to take over and completely determine the value of some variable by adding that variable intervention outside the original system and then putting a direct cause from it into the variable that you've targeted. And so if I have a pre-intervention graph that says education causes income, income causes taxes, and I say contemplate an intervention on income, where if I flip the coin heads, you get 250,000 of income. If it comes up tails, you get 50,000 of income. Right? I can model that by saying, here's the I to represent the intervention. It goes into income, and it annihilates the causes of my target, but leaves intact the effects of my target. And so that's the hard intervention where I actually change income with that coin flip. Clear, everybody? Everybody with me so far? Okay. Now, another kind of intervention that we've studied, but we won't talk about a lot today, is what we call a soft intervention, in which we're going to do something to your income, like add 50,000 if it comes up tails, right, and then add 100,000 if it comes up heads. But the influence that education would have on your income, right, is not annihilated. It's just slightly changed. So those are called soft interventions, and we model those by putting a variable outside the system with an arrow into the target, but we leave in place the original causes that were in the system because we have not completely taken over and determined the value or the probability. We've just changed the way it responds. Okay? So that's a soft intervention. All right, so now I'm going to actually show you an example where I do this onto a system, and then I'm going to ask you to do it as a little exercise. So here I have a system, and this is what we call the pre-intervention graph. These are the relationships among the variables before I go ahead and intervene. And now I'm going to consider a complicated intervention where I do a hard intervention on x1 and x4, and I do a soft intervention on x3. And then the question is, what happens to the system after those interventions? So I start with the same graph, and then I put in the interventions, and I represent hard interventions with I, and soft interventions with S. And then the intervention on x4 eliminates the x2, x3 edges that go into x4. The intervention on x1 would eliminate any effects going into x1, but there are none. And the intervention on x3 that's soft leaves everything intact. And so this is the post-intervention graph for this manipulation. Okay, take the same example and now consider a different intervention where I do a hard intervention on x3 and a soft intervention on x4 and x6, and write down the post-intervention graph. The arrow's gone from x1 to x3, okay? Good? Everything else stays the same? Yeah, the hard intervention is one in which you assign treatment for a variable, as it were. So all randomized clinical trials, right, their intent at least, is to, on the basis of some randomized device, you will be assigned a treatment. Now, whether you actually do the treatment, right, is a separate question. So there's a chain, right, but the intent to treat is assigned randomly. Okay, so now I want to sort of show how you use this in a totally simple example that I've just showed, right, in how association comes from the pre-intervention and the post-intervention system. So here's the age causes both hair color and endurance in the pre-intervention graph, right? And in this graph, hair color is associated with endurance, right? And I won't go into all the rules about why, but we represent that with the sign for independence with an arrow with a line through it saying it's not independent. So this is a way to represent that hair color is associated with endurance, according to this graph, which says that age is a common cause of hair color and endurance, and when two variables are effects of a common cause, right, there's an association. And if two variables have no causal connection, right, then there is no association. And so if I now consider the case where I've painted the hair color, right, with some intervention, right, so I paint your hair gray no matter what it is, right, that X's out the relationship between age and hair color, right, because it's a hard intervention in this case. And so now in this post-intervention system, hair color is independent of endurance. And I'm putting a little m there to represent that this is a different distribution. It's a manipulated distribution. So in the resulting distribution, hair color and endurance are no longer associated, which is why if I paint my hair and I don't know what it was before I painted it, right, it won't give you any information about my endurance. But if you got to observe my hair, it would give you information about my endurance. So I should have mentioned that when I write down a graph, right, that is my hypothesis about the world. It's not necessarily true, but representationally, those arrows represent direct causes relative to the system I'm measuring. And so I'm now saying, I'm claiming that that's an accurate representation. I don't think hair color has anything causally to do with endurance. So when I say one thing is a direct cause of another, that does not mean it determines it in any deterministic way. It means it changes its probability. And then the extreme case of that is it changes it to 1 or 0. But probabilistic causation is perfectly suitable with these kind of representations. In fact, it actually goes better. All right, so representing the causal structure, we've done the qualitative causal structure, we've represented manipulations and interventions, and now we're going to attach probabilities. So in the program, right, we represent the qualitative part of the causal structure with this graph box, and then we can parameterize these boxes, right, in many ways, and I've shown 2 of them here. We can parameterize them as a discrete valued Bayes net, and we can parameterize them as a structural equation model where the variables are continuous and they're functions of their immediate causes plus error. And we'll go into each one at a time. And then once you've parameterized a graph, right, then you can actually instantiate the values of those parameters by putting in actual numbers. So on this level, it's a parametric family, and on this level, it's an actual fully determined statistical model that represents a particular population as well as a response of that system to interventions. So far, all I'm talking about is representation. So if you're trying to represent what you believe to be true of the world, and I say, here are the variables, what do you think are the causal relations among them? Your task is to draw arrows. Sometimes there is an arrow, sometimes there isn't. Right? And then somebody else in the room with the same set of variables might have different beliefs about what's true and would represent a different causal hypothesis. So these arrows, right, are really logical relationships, not necessarily causal ones. So the graph structure that this constrains the model that you parameterized here, and it constrains it in the way you parameterized here. And the parametric model that you specify here, right, constrains which parameters you have to actually put in to instantiate those models. So this is a logical set of relationships, not necessarily a causal one.
So if I could just add one thing to what Richard said, and this is very confusing for people that are starting. These arrows should really not look like causal arrows. This is a workflow. This is like a visual programming system, and this is the workflow in the programming system of how it's operating. So don't view this as a causal graph. View this as a program. And within it are going to be causal graphs that have a structure and parameterization. Does that help?
So you're basically saying each box inside the box is a causal graph, the outside of the box or the abstraction above it is a workflow. What's happening at the causal graph level is
influenced by that workflow.
Be influenced by the workflow, but the workflow itself isn't representing anything causal.
No, and those arrows really need to look different than causal arrows.
These arrows are sequences of.
That's a sequence. That's a sequence of operations. So this has a visual programming workflow, basically language that's up there.
So I can take a graph, which is a qualitative object, and I can interpret it, right, in many parametric ways. And so this parametric model is constrained by this graph. And then once I parameterize the graph, and I want to put actual numbers in it, I'm going to go through this so you can see how it happens. This is constrained by this is constrained by this. So these are workflow constraints. But Greg's absolutely right. It's a poor choice of visual representation that we've just gotten used to. Yeah?
So when you link them like that, with all these workflows, and then you say that's a parameterized model and an instantiated model and whatnot, does the tool automatically copy what's in the root model and then copy it over there so that
Yes.
you can go into the parametric
Yes.
model?
So, and I'll show you this in a minute. The parametric model I built here is constrained by the graph. If I change the graph, I will have to change these parametric models under it. And that will make you do that.
So the tool propagates the changes.
Correct. Or it warns you that it's going to, and if you don't want to do that, you have to say cancel. Okay, so the first parameterization is quite standard and really been studied for 30 years, 40 years now. It's called causal Bayes networks. It essentially says if you give me the graph, and each of those nodes in the graph is a variable, and in this case I've said these are all binary variables, so age is either got a value of young or old, hair color dark or gray, endurance good or bad. That's a simple system with all the variables are just binary valued. And then I say the essential constraint on the probability distribution this graph imposes is that the joint distribution factors according to the graph. So this joint distribution among age, hair color, and endurance is going to factor so that it's a product of conditional distributions where each distribution is now a variable conditioned on its immediate causes. And so with the probability of age conditional on its immediate causes, it has no immediate causes. It's exogenous. And so I just use its marginal distribution, right, in the joint that I'm factorizing here. When I look at hair color, I have to condition on its immediate causes, which are only age, and so I put hair color given age. And when I do endurance, its immediate cause is age, and so its conditional form is endurance given age. And so that is the way I take the joint distribution and factor it according to that causal graph. And that's the essential, right, representational trick to add probability to causal graphs.
So they multiplied then on the right?
Yes. So if I'm going to calculate the joint, right, and I put in what's the probability of A equaling young, right, and HC equaling dark and endurance equaling good, right, that's one of the 8 possible states in that joint table. I would look at the probability of A given young, the probability of hair color equaling dark given age equals young, and the probability of endurance equals good given age equals young. And so that I'd multiply those 3 numbers and I would come up with the probability of the joint state. So in a parametric way of thinking about this joint distribution is a function of parameters, theta. How many? Well, it's 5 in this case. And so here's the full parametric form of a parametric model. The probability that age equals young is theta 1. The probability that age equals old is 1 minus theta 1. Right? And so these, right, 10 rows with 5 parameters give me the whole distribution I need. Now, you might notice that there's 3 variables, and usually you need 7 parameters to encode a joint distribution among 3 variables that are binary. And the fact that you need only 5, right, means that there's an independence constraint somewhere in this graph, or somewhere in the distribution that this graph has forced us to encode. And there is one, and I'll get to it a little more later. But the basic idea is that because of this being the structure, hair color and endurance are independent when I condition on age. Okay, so now in this same format, in this same language, how do I calculate the effect of a hard intervention? If I take this model and I write out the joint, and I say, well, now let's consider, right, painting somebody's hair gray or dark. Well, I do the hard intervention, I X out that edge. How does that translate into the new manipulated distribution? Well, I'm representing that with a P sub m, and it's also got to be a joint among those 3. And the first thing I do is I take the term for hair color, conditional on age, which was required by this particular edge, and no other edge is going into hair color. And now this edge is gone. And so what do? I replace that term with the probability of hair color given my hard intervention. And so in the simplest case, I might say the hard intervention is to paint everybody's hair gray, right? And so then the probability of hair color equals gray given the hard intervention would be 1. Or I could say flip a coin. And so it might be that the probability of hair color given the hard intervention, right, is 50% gray, 50% dark. I have control over that. But the only thing that the probability of hair color depends on now in this new system is the hard intervention I've increased. And what about the other terms? The probability of age and the probability of endurance given age. They come down identically. So by factoring this distribution, I've modularized it. And so I've located exactly the term in this factorized joint that has to be replaced, and I've left the other terms that don't have to be replaced identical. So now I can completely determine the manipulated distribution in this very simple way. And in a soft intervention, it's similar with one little additional trick. So if I'm going to replace the hair color given age with the soft intervention on hair color, what is this new term going to look like? The probability of hair color given what? So it's just soft in your version, but you're saying soft and age. Yes, that's correct. Because we're still allowing this edge to exist, and now you have to express this guy as the conditional distribution of him given all of his immediate causes, which now includes soft and age. In the hard case, we eliminated the age as being an immediate cause, and so it just had hard here. But now that we're doing a soft intervention, age remains as a variable that is an immediate cause. And the other guys come down just as is. The soft intervention just adds a node in a particular place. And so the idea that it's exactly in that particular point in the distribution is the key to it. But the difference is in the hard intervention, we eliminated age, and now we keep it in. And that's it. That's really, you can do an awful lot with just that simple set of rules. Okay, so now I'm going to go back to Tetrad and actually show you how to actually parametrize, right, some of these models. And then you should follow along with me, right? And I'm going to do it for the discrete valued Bayes net first. So I take my graph, which has age, common causing, gray hair, and endurance, which is this one right here. And I'm going to say, put a parametric model on the workbench. And then I'm going to add this, which is that thing that Greg said shouldn't be represented as a blue arrow, but it is. Right? We use the same editor underneath it. Right? And then we have the graph, right, constraining or workflowing into the parametric model, but we don't have a parametric model yet. We have to choose which one we want. So I double click this box. Now I have a pull down that gives me several choices. Bayes, SEM, or generalized SEM. And so I'm just going to do the Bayes, and then I'll say OK. And I'm going to say set up manually so you can see how it works. OK. And now for each variable, it says, well, how many categories and what are the values for those categories? So if I say age is two-valued, right, I might say old and young. And now I've set up a system so that age is a binary variable with values age equals old and age equals young. And if I click on this guy, same thing, right? And I can go to the presets button and click that, and I have options that I can do that are easy. So if I say yes, no, that will be for that. And endurance, I might say good, bad. And that's my parametric model. So no numbers yet, but I've set up the structure of the variables and how many values they have. And so now the parameters I then have to add as numbers are now determined. Right? And so if I change this graph, then I will have to change the parametric model under it as well. Now that I have that, what if I want to put in actual values for those parameters? Well, I put an instantiated model on the workbench and then drag the arrow into it. And now that instantiated model is going to be constrained by the parametric thing I've set up. So now, what probability do I put on age equals old and age equals young? Well, I don't have to consult, right? I can just make this up because I'm playing God here. I'm building a representation that is the truth insofar as the program cares about it. So I say 0.5, and if I hit the tab, the program is smart enough to know that these have to add up to 1, and so it fills in the rest for me. Now, if I click on gray hair, what's going to be the difference I see in terms of what I have to do? I have to do a conditional distribution, and so I have to put in probability of gray hair equals yes or no for each particular value of age. And this is what expresses the causal strength or the causal relationship quantitatively. Okay, so let's do it. So the probability that my gray hair is equal to yes if I'm old, let's say, is quite high, so let's give that a 0.8. And now the probability that my gray hair is yes if I'm quite young is low, and let's give that a 0.2. So the difference between 0.8 and 0.2 here is the causal strength of that arrow in quantitative form. That's expressing how strongly age actually influences gray hair. But to your question in the back, it's probabilistic. Right? Even if my age is old, that does not determine whether I have gray hair or not. It just makes it much more likely. Okay? And if I do the same thing with endurance, right, the fact that my endurance is good, if I'm old age, maybe it's 0.4. And if I'm young, maybe it's 0.8. So in this case, it's the same sort of thing, but it's less of a difference between the age equals old and the age equals young. And so that's how I parameterize a conditional distribution according to a causal graph. But this parametric model structure, right, is a general way to interpret this graph, but you can interpret that with an infinity of different instantiated models. Each instantiated model is a different population, but what do the populations have in common? They have in common that in every one of these populations, gray hair and endurance is conditionally independent on age. That's going to be true of every single population I define here. I cannot avoid, in any of my instantiated models, I cannot express a population in which that isn't true. Because that's what that graph constrains me. Now, I could take a sample from those populations in which that is not true in the sample, but I can't define a population, right, from this parametric model in this graph in which endurance and gray hair are not independent on age. So that's common to all those populations. And that's the key to figuring out something about the causal structure, because I'm going to find out what constraints hold in the data and then say, well, what are the causal structures, right, as graphs that would get me that constraint? And that's how I get all the way back. If I know the causal, if I specify a causal structure and I say, given the parametric model that I specify for that causal structure, and give me data, then I can estimate the instantiated model. And I'll get to that in a minute. I'm going to show you that exactly. I want you guys to understand estimation. So what I'm doing here now is I'm saying, well, I have an instantiated model that defines a population. Let's pseudo-randomly come up with a sample. So I could do that by creating two different samples for the same instantiated model, because it's like taking two different samples from the same population, right? And I can say, okay, simulation 1, right, I didn't set it parameters, but this is actually giving me 1,000 data points. So now it's generated pseudo-randomly, right, 1,000 data points from that population. And so now, if I take a different simulation, I'll get a different 1,000 data points. And here, I might get a different 1,000 data points, but from a different population. So I'm sampling a different population. But what do all these populations have in common? They all came, or they're all governed, by the same qualitative causal structure and the same parametric family. Different parameter values. So this is a different population than this. This is two different samples from this population. And this is one sample from this population. And all those populations are constrained by the qualitative causal structure that is the root of that tree in the graph. And then what I'm going to do is take these simulations and run search algorithms on them to see what I can find about the structure that generated this data. And then once we understand that, then you'll put in real sample data from what you've done as experimenters or observational studiers, and then we'll apply the search and we'll see what we can learn. This is the other parametric form that's very common, and it's very, very easy to work with. And so I'm going to show it to you, and all the examples, or many of the examples that I'm going to work on later, are going to follow this parametric family. So now consider three variables, same causal structure in terms of the arrows and the shape, but now I have education, income, and longevity, and I'm going to consider these as continuous variables, right? So I can define the causal relations parametrically now, right, for each variable by assigning it a function of its immediate causes plus noise, right? So x is going to equal some function of the immediate causes plus an error term epsilon x, and then I have to assign a distribution over the error terms, and then I have a full statistical model with a full population specified, right? And so for this graph, right, the path diagram that usually accompanies this, right, has linear coefficients beta 1, beta 2, and the error terms that are represented, right, as extra variables that are in the system but not as part of the original representation. Yeah?
What do you do with the parameter may work both directions, like education affects income and income affects education.
I can assign parameters in that, but that would be a different causal graph.
You can go both directions.
Yes, I can go both directions, and I can add both arrows at the same time and build a model on that basis.
That wouldn't be a separate graph altogether?
That would be a separate graph altogether.
It would be.
Yes. Yeah, I mean, if I have an arrow going from education to income, that is implicitly telling us that income has no effect on education. That's what this graph claims. So for you to be right about that, income, because you would have to draw a different graph. So I'm representing the hypothesis that, right, income has no effect on education.
So you can't have subgraphs.
Or different graphs, different hypotheses. And in fact, it's a complicated matter because you can conceive of these things as representing a population at a moment in time, right? And so if I say for a particular person, right, if I change the level of their education, what would that do to income? Well, you'd really have to think about the income as in the future, right? So I'd have to go back counterfactually and say, well, had they been more educated, would they at this point in time, at age 30, for example, would they have more income? Well, the causal structure here says, yeah, they have a different probability, right, for whatever income they have, and so they might well have a different income. But if I'm looking at the thing as an evolving process, and I say, well, sometimes people get educated, they make money, and then they decide I have more money now, I want to get more education. They go back and get a master's degree, they get more money. They said, this is too much fun, I want to get a PhD. And then they make less money. No, okay. Etc. And so on and so forth. Okay. So the equations that correspond to this path diagram are listed here, and the only difference that I want to make you make sure you see is that this is education colon equal, because that's an assignment operator. It's not an algebraic equality. So I can assign a value of epsilon education, and that'll assign whatever it has to education. The same thing, education here, beta 1 times that plus its error term is what I assign to income. But if I change income, I don't change these guys necessarily. And that is that assignment operator, like in computer science, which encapsulates the asymmetry of the causality, which is extremely important, because when I was doing this at first, they didn't use that notation, and it was very confusing. Because it looks like if income is equal to beta 1 times education plus income, I could just subtract it from both sides, I could switch to which side it was, and it would look like it was the causal structure was reversed. So that's not what we're saying here. And so in matrix form, the structural equations are given this way. And then what I have to do to finish out the idea is put an exogenous distribution in over the exogenous, distribution over the exogenous variables, right? So I might say the joint probability of epsilon, education, income, and longevity is, right, pairwise independence because they're not connected with any structure, right? They have a multivariate normal, and every pairwise entry in the covariance matrix is 0, and every variance is non-zero. So that's a simple way you could do it. Right, so that would be an example, and that's the typical distribution that's used in structural equation modeling. Right? Error terms are considered to be normal, right, with mean 0, right, and some non-zero variance. And that follows in a certain way because of the central limit theorem. What do these error terms represent? All the other causes I've left out, right? So if someone, right, is exposed to radiation from asbestos, and I say, well, what's the value that they're going to have for lung cancer, right? There's lots of other causes that might be needed to actually fully determine whether they get lung cancer or not, right? And if you add up all those other causes and you put them into a single term called the error term, right, then that gives you, right, that extra variable. And if you add up a set of independent things, they're normally distributed with mean 0. Okay? All right, so now I'm going to go back to the program and then to show you how to build, right, those form of parametric models, and then we're going to sort of switch gears and we're going to go to search. Okay, so if you look at this graph, right, which has x1 causing x3, x2 causing x3, x2 causing x4, and I say put a parametric structure under it, and put a parametric structure under the first graph. If I look at the first graph and I say, what's the parametric structure if I give it a SEM, structural equation model, it just looks like this. where everybody has a linear coefficient attached to an edge and an error term. And so there's nothing to actually build in terms of what tables and it's determined by the structure of the graph once it's a structural equation model. It looks like that. And in this case, if I did it as a SEM, it would look like this. So it's just sort of building the path diagram from the causal graph to give it the structure of that parametric form. And then for me to instantiate these parameters, I have to put distributions on the error terms. They default as mean 0 and normal. And I have to assign values for these linear coefficients. That's what it would be to instantiate these models. So let me just show you one. So if I say instantiate the model with the x's, oops. Now there's one choice that's called a standardized SEM instantiated model. So that's going to set the thing up so that all the variables have mean 0 and variance 1. Right? You just take out the scale into everything, and you can always, much easier to interpret everything, and all the stuff is easier. So in that setting, right, it defaults by just plucking values out of the air and putting them in there. But if I want to change that, I can click on them and then type in 0.5. Right? For this one, type in 0.6. And this one, type in, oops, 0.7. So now I have defined a standardized model in which the correlation of x1 and x3 will be 0.5, x2 and x3 will be 0.6, x2 and x4 will be 0.7, and those also correspond to causal coefficients. And so if I raise x with an intervention by 1 unit, then I will have made x3 raise 0.5 units in expectation. Not necessarily raise it, but its expectation now will be up by 0.5. So these coefficients tell you how much you're going to change the expectation of the effects. And generate data, it tells me, oh, how many data points do I want? Right, I'm getting 1,000. If I say give me only 10, I can do that too. I say simulate. And now I have 10 data points, but you'll notice these are now continuous variables, right, that always have mean 0 and variance 1.
We're going to go to the third section, which is, we've basically been at this point talking about the representation itself and some of the basics underlying it. This next part we're going to talk about searching over that representation when you have data and background knowledge, being able to build models from data, which is more the discovery part of the title of this workshop. So there's really 3 main components that we need to learn causal graphical models from data. One is we need a model representation, and as I said, that's what we've been talking about so far. Another is a way of searching over those models, looking at different kinds of models. And then a third is being able to evaluate those models relative to how well they're supported by the data that we have and any background knowledge that we give the system. So in terms of causal graph search, you can imagine if we just had 3 variables, there's a possibility for searching over many possibilities. This is third one is y is causing z, here x is causing y, and y is causing z. The system has some way of searching over these different possibilities and evaluating them relative to the data and background knowledge. And of course there are a huge number of these models. So let's just look at that for a moment. What if we just had one variable? How many models, causal structural models, are there? Just the structure. Well, there's just one. There's no arcs to be had. If we have 2 nodes, then there's, and we're not even considering latent variables here, just assume there's no latents. If we have 3 variables, x and y, x can cause y, y can cause, or y could cause x, or there could be no causal relationship between them. So those are the 3 possibilities. When we go to 4 variables, turns out there's 543 possible causal graph structures. And here you can see how it escalates super exponentially. So even if we had 7 variables or nodes, we have over 1 billion possible causal models, which is, and then to 10, it's over 10 to the 18th. So we need heuristic search in general, and a common type of heuristic search, and we're not going to have time to go into the details, but it's to use some sort of greedy search. And some of the algorithms we use will certainly use those. One type of greedy algorithm is to do a forward search, starting with an empty graph. The nodes have no arc between them, and add arcs in a sort of a greedy way that you add the arc that most increases the score, and you get to the point where there's no arc that you can add to the current structure locally that will improve the score, and then you do a backward search trying to see if there's arcs that you can remove singly that increase the score, and you continue that until there's no improvement in the score and you stop. And these kind of algorithms are often fast, this kind of greedy search, particularly if the network is not dense in terms of the data generating causal network or causal model, causal system. But of course it only searches over a small portion of all possible models, so there's no guarantee that you're necessarily going to find the causal model that's generating the data. Also, another dimension is the data itself, and we can have, and Richard talked about this, we can have experimental data which involves interventions, and RCTs are a classical example of that. We can also have just observational data, like electronic medical records would be a good example of that, where we don't typically have any interventions that were done under controlled conditions. It's just observational data about what happens in the care of patients. And of course we could have both of those. Some of the data could be experimental and some could be observational. Turns out that there are algorithms that exist that can work with just observational data. There are many approaches that could use experimental data, and then there's algorithms that can use both experimental and observational data. But for the rest of this workshop, I'm just going to focus on observational data because that's the least obvious type of data from which we could discover causal relationships, and therefore I think the most interesting for discussion. There are 2 main types of algorithms that have been developed for learning causal graphs from data. One is based on constraints, and I'll talk about that in the next slide. And the other is called score-based, and it's based on scoring. I'll talk about that also in the next slide. So constraint-based, what it's doing is it's finding a causal graph that's consistent with a set of statistical tests. So for example, if you had discrete data, you could use a chi-square test on the data and find independence relationships or conditional independence relationships among the variables in the data, and those form constraints. All right, so you have to have some sort of thresholds that you're using, but if you are alpha values, but if you make a commitment to that, you end up with some constraints. X is independent of Y given Z, according to a chi-square test. Here's the most simple, one of the most simple examples we could imagine, which here's a causal graph. X is causing Y. And the constraints that are found so far in the data is there's a dependency between X and Y. Well, then this model, X causes Y, is consistent with that constraint. If in fact there had been an independency in the data between X and Y, that would be inconsistent with that causal model, because we would expect X and Y to be dependent, given that X is causing Y. We'll talk about constraint-based more in a minute. Score-based, and one of the main types of score-based models are Bayesian models. They assign a score to a causal graph, and according to how well it's supported by data and background knowledge. So if we go back to our example with X causing Y, the Bayesian score for this would be the probability of the data, given that we have this causal graph, X causing Y, times the prior probability of the causal graph that X causes Y. And many times algorithms, users will decide to make the prior probability some sort of non-informative prior or uniform prior over the causal possibilities, and have the data kind of drive the score. But in any case, that product there on the left is proportional to the posterior probability of the causal graph given the data. So when we're searching over this product, which is the score, we're essentially searching over the posterior probability of the causal graphs. We're not going to have time to go into the Bayesian in more detail. We're going to talk more about the constraint-based just because of the amount of time we have, but I will give you some references that talk more about the Bayesian approach if you're interested in learning more about it. I also want to briefly talk about latent variables. So a latent confounder is an unmeasured node or variable that causes one or more measured nodes. So here I'm going to represent a latent variable with a white unfilled node or a circle. And H represents hidden here. So what this is showing is that we have a hidden variable, we haven't measured it, and it's causing a measured variable X and causing a measured variable Z. Y, sorry. And in Tetrad, a latent confounder is represented with this kind of edge notation where we have a double-headed arrow between X and Y. Okay? So Tetrad contains algorithms, for example, the so-called GFCI algorithm that searches for causal graphs that may contain latent confounders. So it does represent that, it does model that possibility. And it represents an equivalence class of causal graphs using what's called PAGs. Those stand for basically, I'll go back into that in a minute, but what those PAGs contain are different edge types. It contains that there's no causal relationship between a pair of variables X and Y, or that X causes Y, or that Y causes X, or that there's a latent confounder of X and Y. This is really representing this little white circle represents that we're not sure if there's an arrowhead there or not. So in this case, it could mean that either X causes Y or there's a latent confounder between X and Y, and we're not sure which one it is. So sometimes the algorithm can't make that distinction. This is sort of the related relationship. This, where we have two circles on each end of the edge, means that we're even less sure about the relationship. We know that there is some relationship between X and Y, but we're not sure if X causes Y, Y causes X, or they have some sort of latent confounder, or maybe there's a latent confounder and there's a causal relationship. So those are, PAGs stand for partial ancestral graphs, and again, because of time we won't have time to go into that, but I want to give you just a sense that there is a representation for these possibilities within some of the algorithms in Tetrad. There are also algorithms that assume so-called causal sufficiency, and what causal sufficiency means is that there are no latent confounders among the variables. That's what you're assuming. And some of the algorithms in Tetrad assume that FGES is one of them. And so what Tetrad does in that case is it has the following kinds of edge types that are called patterns. Again, it could be that there's no causal relationship between X and Y. X could cause Y, Y could cause X, or you could have this kind of edge that has an undirected edge that basically says you're not sure if X is causing Y or Y is causing X. Okay, but one of the two is the case.
Okay, so now I'm going to give you the demo of how Tetrad does these searches for patterns or PAGs in the context of a simulated example where we know the truth, right? And I've already sort of built it for you already. So let me go to that system and have you follow along. So in this case here, I created a structure in which x1 causes x3, x2 causes x3, x2 causes x4. I parameterize that as a structural equation model. So I'm going to take a graph and build it to have that structure. And what I'm going to do is run two kinds of search on the data I simulate from this graph and then see what we get and how it relates to the truth. So I'm going to parameterize this. I'm going to put an instantiated model in place. And I'm going to simulate data from this. And then I'm going to do a search. Okay, So the graph has to be interpreted with a parametric model. I have to instantiate parameters. I have to simulate a sample from that, and I have to put that sample into the search and see what I get out. So let's do it as a SEM. Easy enough. And let's do it as a standardized instantiated model. Easy enough. All right, let's make everything positive just so it's sort of easy. And now let's simulate data. So I have 1,000 data points, which gives me plenty of sample. Say done. And now, when I try to search for what is the causal structure that might be generating these data, the search algorithm will only pay attention to these data. It will not know what's behind the scenes here. So in some sense, these three boxes, which I have created as if I'm playing God, right, are not available to the search algorithm. And so we're now going to find out what the search thinks might be generating this graph. So if I do the search, I get all these possible algorithms, and that's too many to think about. And so on the left-hand column, you have options for restricting the sorts of things you're searching for. And so if it's the simplest case, let's forbid latent common causes. Let's imagine that we're arrogant enough to think that we've measured all the latent common causes that are out there. So this is the unrealistic case, but nevertheless, we can do it. And I can choose the algorithm that Greg mentioned, fast greedy equivalence search, and then say set the parameters. And I'm just going to choose the default parameters and say run the search and generate the graph. And this is what I get. Now here's a handy feature you can right away use in Tetrad. If I go to the original graph, I can right click in this space and it says layout, and I can copy the layout. And then I can go to here, right click, and paste the layout, and it'll put that structure in the same spatial orientation as the graph I generated. So what do you notice about the output of the search on the right compared to the truth that generated the data on the left? Right. So it got x1, x3, nailed that one. x2, x3, nailed that one. Then x2, x4, right, is an undirected edge. Well, it turns out that if the program only has the independence relations true in the population from which we sampled, that's all we can get. There is no more information in the sample to orient this particular edge. And another way of saying that is, if I flip this edge the other way, the model, right, would have to be reparameterized and generate data. But the statistics of that model in terms of how it fits the data would be identical to how this model would fit the data. There is nothing that would distinguish that model from this one. And the way that's represented in the search is by saying, well, there's an undirected edge. We can't tell which direction it's going. Nobody else could either. And so this is all the information we can get out of the data, right, given the assumptions we've made. Now let's do the same thing, but now let's not be so arrogant, and let's assume that we actually have the possibility of latent confounders. So I'll do this search here, but this time I'll click on Allow Latent Common Causes, and I'll do the FCI, which is Fast Causal Inference. Set the parameters, run it, and again, copy and paste the layout. And now I have something quite different. This is the true causal qualitative structure from which I parameterized a SEM, instantiated the parameters, simulated a data set, and then take that data set and then put it into the search. But now I've allowed the possibility for latent common causes, and now what do you notice? There's a lot less information about the causal structure which might have generated the data in this output. This edge now from x1 to x3, what does that mean? Either x1 causes x3, there's a confounder. But what is not allowed is that x3 causes x1. So that's eliminated from consideration. So I've learned something quite important. Negative causal information, which is all I can learn in this case. Same thing here. So x3 is positively only a sink. It cannot cause anything else in this set of variables. the relationship between x2, x4 now, whereas it used to be it's either one causal direction or the other, now it's either x2 causes x4, x4 causes x2, or there's some latent confounder. So we really can tell very little about the relationship between those two variables. But there's lots of other things that you can tell. This can tell you for sure that x1 and x2 have no direct relationship, and in fact, right, x1 can't cause x2, because if it did, it would have to go through x3, and it can't, and vice versa. So we know that x1 can't cause anything except for x3, x2 can't cause anything in x1, it might cause x3 and x4, so we know a lot from this structure, but not nearly everything we would possibly want to know. Yeah. Okay, so now I'm going to take this entire structure, right, I'm going to copy it by just surrounding it here and saying Control C, and then Control V to copy it. So now I have the same thing, but I'm going to do one thing more. I'm going to go into the graph, and I'm going to add one more variable, x5, and that's going to be an effect of x3. So it's the same structure I had before, but now I'm adding a variable. And if I say done, well, the program's going to tell me, well, you just modified the graph, and so you're going to have to modify the parametric model and everything else downstream of it. And I say, yeah, go ahead and just execute that. In which case, it's going to arbitrarily pick parameters, arbitrarily sample right here, and then find in the search algorithm something that looks like this. Let me go back and copy the layout so it's easy to see. So what do you notice here? On the left is the truth, and on the right is the output of the search when I'm assuming no latent common causes. And you notice that everything looks the same on the top part of that model, but now the x3, x5 is also oriented down. But it oriented x1, x3, and x2 at x3 as well. Right? So this is what I get with the assumption of no latent common causes. But now let's do the same search. So what does that say? Again, it says that x1 and x3 might be related by a confounder, or x1 might cause x3, can't tell which. x2 causes x3, or there's a confounder, can't tell which, and x2 causing x4, it could be anything. But the x3, x5, that's fully oriented. And what that means is it's detectably unconfounded. You could now do it. In this particular case, we were lucky. I wasn't lucky. I knew this was going to work. All right? That's why I'm teaching the course. And in this particular situation, the search algorithm can tell that there's an edge and it's unconfounded. And it's doing that because it has, in some sense, found detectable instrumental variables. For those of you who have ever seen instrumental variables, right? x1 is related to x3, it's into it, it's got no direct connection to this, and it's got to be independent of any of the confounders that might actually be influencing x3 and x5, and it turns out there are none, and it can detect that in this case. Okay, so now you've seen how this works. I'm not going to do estimation. This is going to take way too much time. We can come back to it. But now let me just give you a couple slides on what is behind under the hood that allows us to do this search and discover something about the structure, but not everything. Well, what we're doing is we're eliciting testable constraints that we can look at in the data that are implied by just the qualitative causal structure. Nothing parametric, nothing quantitative, just the causal structure of the actual model. And the basic axiom that we actually assume is called the causal Markov axiom. In a large set of circumstances, namely directed acyclic graphs, that's equivalent to D-separation, which is a relation Pearl and his students discovered and articulated way back in the 1980s. And those take a causal structure without numbers and then give you a testable prediction in the form of independence and conditional independence constraints. So now you're having qualitative structure, a qualitative relationship. There's no numbers. These are all non-parametric. And those bridge principles would allow us to go the other way. If we have the testable constraints, we can figure out what are all the structures that would entail those constraints, and then build a representation about what's common about all those structures. And those circles and those arrows and those double-headed arrows, that's the common to all the structure representation. And to give you a very simple picture of the very, very simplest assumption we could make to get this all off the ground is to start out by saying, well, what if two variables are causally disconnected? If V1 doesn't cause V2, indirectly or directly, if V2 doesn't cause V1, and if there's no common cause of the two. So that's what it is to be causally disconnected. The weak causal Markov concession says if two guys are disconnected causally, then they're independent probabilistically. And that's it. This is the key assumption that gets everything else off the ground. So this assumes that if two variables are probabilistically associated, then there's got to be some causal explanation, right? They must be causally connected. One causes the other causes the one, or there's some common cause. So if that's something you're amenable to, and then we have general structural equation models, those entail the Markov axiom, which says that every variable is independent of its non-effects, conditional on its immediate causes. And deseparation comes from the Markov condition and acyclicity. So deseparation is really the thing we use. You get a causal graph, you show deseparation, you compute deseparation on any variables and a set of conditioners in that graph, and that tells you what's independent of what. In the population, any population that graph could parameterize. So if I give you this graph down here, and I say you build any parametric model you want, you instantiate that with any level of values you want. I don't care. That population, right, will contain these independencies and conditional independencies, just by the structure of the graph alone. And then the search procedure goes the other way. Starts here and goes back that way. Right, so in this case, if I observe that x is independent of z given y, and I assume no latent confounding, well, what I get is that there's three structures that all entail this independence by deseparation of the Markov condition. So that's all I could know about what's causing what if this is all I observed. So you learn something, but you don't learn everything, and that's just the best you can do, right, without doing further experiments. And now Greg's going to take over and go a little bit deeper into the constraint-based side.
Richard has covered the kind of general principles that exist between connecting independencies that you find in the data with independencies that are being expressed in a causal model. And what I'm going to do is cover an example that I hope will bring this home and illustrate it in a very concrete way so that you understand more deeply what's going on through sort of induction on that example. So just to cover one more time the basic idea, in the constraint-based method, what's going on is we determine the constraints that hold in among the variables or nodes based on statistical test, and then we evaluate causal graphs that we're searching over to determine if it's consistent with those constraints. And we find those that are. And among those that are consistent with the constraints, we output a set of causal graphs, or really what we output is an equivalence class in that language that you saw before. So let's go through a concrete example that I think will make this clearer. So let's suppose that we have a really simple system for illustration. We've got three binary variables, x, y, and z, and the following is known. X occurs before y, and x occurs before z. And that just makes it a little bit easier to give the example, but oftentimes we do know those kind of temporal relationships. For example, in EMR data, we know that a particular drug was given before a given test was obtained before a given outcome occurred. So it's not totally unrealistic kind of assumption. And then let's suppose that X, for example, could be mutation status of a gene, Y could be the expression level of some other gene, and then Z is the disease status for some disease that we're interested in. And our question is, does Y cause Z? Okay, does the gene expression of a particular gene Y influence causally disease status of some disease of interest? So let's suppose that in the data, then we've got a lot of data, it's really good data, and we get these dependencies. We get these four constraints. We find that first we find that X and Y are statistically dependent, pairwise, and Y and Z are pairwise dependent. X and Z are pairwise dependent. So we've got all pairwise dependencies among these three binary nodes. And interestingly, we also find that X and Z are independent given Y. So that's what we found in the data so far. And let's consider the following causal graphs. Okay? Search is going on. The system is searching over causal graphs. This is one that it's considering right now. The question that it's asking itself is this causal model that I'm hypothesizing consistent with those four constraints? The data is telling us that Y and Z are dependent, but in this causal model, there's really no way in which that can happen. So this model is inconsistent with that second constraint. What about this one? No, for a similar reason. This one is between X and Y. We found in the data they were dependent, but here there's really no way for them to be dependent according to this causal model. And I'm calling on your causal intuition here, but we can make mathematically precise all of these ideas, these associations. What about this one where X is causing Z and Z is causing Y in our model? Is that consistent with those constraints?
No.
Okay, so we've got the pairwise dependencies here that are consistent with this model. What about the fourth? So the fourth one is saying, so the no is correct, that X and Z are independent given Y. We're finding that in the data. But here, if we conditioned on Y, we wouldn't expect that to affect the dependency between X and Z. But in the data, we're finding that it does. That in fact they're independent given Y, X and Z. So that's not consistent with what we're finding in the data, the fourth constraint. What about, the computer keeps searching. This is the next one it's considering. What about this one?
Yes.
Yes, okay, that is correct. We get the pairwise dependencies as before between all the variables. And here we would expect that the only way that X can cause Z, have a causal influence on Z is through Y. If we condition on Y, we would then expect them to be independent, okay? And in fact, so that one's consistent. Now, if we had more time, we could go through and show that this one also satisfies the four constraints, even if we have a hidden confounder of X and Y. And even if we have a hidden confounder of X and Y and X causes Y, that one is also consistent. It turns out for the 90 additional causal networks, which we could enumerate if we had time, none of them satisfy the four constraints above. Okay? So what do you find that's consistent among those three that are the same? What's similar among the three that are consistent with the four constraints in the data? Yeah, and all of them, Y is causing Z, and there's no hidden confounder between Y and Z. Okay? So if we're building the equivalence class based on the language that Richard was showing, we would have an arc from Y to Z, and it wouldn't have any circles. It'd just be an arc from Y to Z, showing that Y causes Z in an unconfounded fashion. So what's just gone on? Basically, what we see is that what's happening in constraint-based causal discovery search is it reduces a large number of causal graph possibilities to just those graphs that are consistent with the constraints that we've found in the data. And so in our example, it's reduced it from 96 graphs down to 3 graphs, those 3 on the right, that are consistent with the data. So we got a big reduction. It also looks for the causal relationships that are common across the graphs that are consistent. And the thing that we found that was consistent was that Y is causing Z in an unconfounded fashion. And it outputs that. So that's really the essence of what's going on. The sort of the patterns that it's looking for can be more complicated than just the one in this 3-variable example. But I wanted to show you that actually this paper in Nature Biotechnology in 2013 was looking at epigenome-wide association of data that implicate DNA methylation as an intermediary of genetic risk of rheumatoid arthritis. So it really is very similar to what we just saw, but with some real data. So what their goal is to determine whether specific sites of DNA methylation causally influence rheumatoid arthritis. So they used 354 positive rheumatoid arthritis cases and 337 controls from patients in Sweden. And in their case, X represents a SNP measured with an alumina chip. Y represents methylation positions, DNA methylation positions, again with an alumina array. And Z is the rheumatoid arthritis status, either yes or no. And of course, X and Y are changing. They're trying many different SNPs. They're trying many different methylation positions. Z is always rheumatoid arthritis status. So the search is over different X's and different Y's and looking for the kind of patterns that we saw in the example that we went through. So in going back to our flow diagram, the input is the array measurements, SNP measurements, those are X. The DNA methylation array measurements, that's Y. And the rheumatoid arthritis status is Z. And they're doing a causal analysis very similar to what we just did on the 3 variables and looking at the causal graphs on X, Y, and Z. And they're selecting those X, Y, and Z triplets that have constraints that are supporting that Y is an unconfounded cause of Z because it meets those 4 constraints that we went through before. And those constraints are being obtained from the data. Those lead to candidate methylation positions that are causally influencing rheumatoid arthritis, namely the Ys that support the 4 dependencies. So going in a little more detail, what they found was that 535 SNP DNP pairs satisfied the 4 needed constraints among X, Y, and Z, and that these pairs consisted of 264 unique SNPs and 9 unique DMPs, the differential methylation positions. So these 9 DMPs then are candidate causes for rheumatoid arthritis, because just like in our simple toy example, we found that Y was an unconfounded cause of Z. In this real data, these 9 satisfied those 4 conditions, providing support that those DMPs, namely the Ys, were unconfounded causes of the Zs, namely the rheumatoid arthritis. I just, maybe as a footnote to some of the questions that are coming up about statistical issues, we have explored some of the methods for this. It's not in Tetrad yet, but it should be soon. Basically, we have some Bayesian methods for estimating the probability that all the constraints that support a given causal graph are jointly true. So in some sense, the probability that all those constraints are jointly true is the probability that the equivalence class of graphs is true. Okay, so it gives you a probability. I think that's what you're looking for right now, is you don't want to just know what the answer is, you want to know how confident you can be in that answer. If you have a probability that all those constraints are jointly true, that gives you the probability that the equivalence class of graphs is true. Okay, so there is some work in this area, but we're not going into it in detail here. So they did some experiments in terms of basically looking at another population. So they evaluated a separate set of patient cases to assess whether the methylation differences occurred in those 9 particular DMPs, differential methylation positions, that they found. They only had 12 rheumatoid arthritis cases and 12 controls, so it was pretty small. But they extracted monocytes from whole blood using flow cytometry, and then they measured the DNA methylation levels between the cases and controls, again using these arrays, and they compared the methylation levels for the 9 DMPs across the cases and controls. And the results that they found was that all 9 DMPs showed methylation changes in the same direction as the original analysis. and 3 of them were statistically significant at 0.05 or less. One was at 0.06, one at 0.11. But of course, there's only 24 cases, 12 cases and 12 controls, 24 total cases. So they concluded that this provided some reasonable support for the original study, which was larger. So then in conclusion, basically they found that 30% of the DMPs in the reproducibility experiment had statistically significant changes that were in the same direction as the original study, and that of course additional experimental validation would be needed to provide additional support that these are causal. But this was a study that I just wanted to show you that was in line with the example that we had gone through together. Then I want to just mention also that I just went through 3 variables, but the algorithms in general are dealing with hundreds or thousands of variables, so you get much more complicated relationships, and it's looking for more complicated patterns among the variables that are given by the constraints in the data. But there are some general things that are going on here that I want to emphasize. One is that what they're doing is they're finding general patterns of statistical dependency among the measured variables that are consistent with the causal graphs they output, no matter how big those graphs are. And they make the following assumptions. One, that there's a causal model that's generated the data. Another is that there's the independence relationships in the causal model entail independence relationships in the data. And independence relationships in the data entail independence relationships in the model. And of course, these assumptions can be made a lot more precise. But at a high level, those are the main assumptions that are being made in these constraint-based algorithms for causal discovery. Now we're going to go into and really wrap up the workshop by talking about a couple examples. Richard's going to talk about one having to do with college plans, and then I'm going to talk about one having to do with cancer.
Okay, so here's where we're going to have you guys load in data, right, and then follow along with us in searching for explanations of that data that are causal first. So this is from a study that was done literally in the 1960s and 70s in Wisconsin, when the issue was, what are the factors that might determine whether young men and women make plans to go to college when they come from a variety of socioeconomic strata, and what influence would their parents have on their college plans, especially when you take into account IQ and gender. So we're going to load in the college plans data, and then we're going to enter some background knowledge, and we're going to both do a search for what we can find and also then do a bootstrap so we can figure out how likely we think some of the output features are given that data. So I'm going to go into the program, and I'm going to start a new session by going to the File menu and saying New Session, and that gives me a blank slate. And now if you want to put in data that you have measured or somebody else has measured, you start out by clicking on the Data node. And the data and simulation are sort of the same thing, but data refers to something you've measured and are going to import, and simulation refers to data you've pseudo-randomly generated from a known structure that you've defined. So I put that onto the workbench, and then I double-click it, and then I get an empty data frame, and then I go to the File menu and say, Load Data. And then I have to point the program to the directory where the data is. And so you should have in the data file that we actually gave you over there, a file that's called collegeplansdata.txt. So this is just a flat text file with 10,000 and some number of cases on those 5 variables. And so if I say choose, I get this kind of interface which says to me, okay, now I'm going to figure out what kind of data you have. Is it tabular data or is it covariance data? Well, this is tabular, and this is a preview of what it looks like in the file that it's trying to load. Are the data continuous or are they discrete? Well, I know these data are discrete, and so I'll click on the discrete. And the delimiter, right, for what the data points are delimited. These are tab delimited data, and the first row of the variable names, and the rest I'll just ignore for a second, and I'll say validate. And then I get a window that says to me, yes, there are 10,318 cases, validation passed with no error. And I will confess that even though we've worked on the data loading, sometimes when you have your own data set and you're trying to get this in, it can be very frustrating. And I say load, and then I have a data set that looks like this. So I've got the sex, the IQ, which is a 4-point scale, college plans, yes or no, parental encouragement, and then socioeconomic strata, and I have 10,318 cases. God bless you. So now before I do a search on this data, I want to actually encode something I might know about the data and the variables that are under study. And so to do that, I click on the knowledge box and put that up on the workbench, and then drag the data into the knowledge box so it just tells me what I'm going to have to specify knowledge about. And then the first option that comes up if I double click knowledge is tiers and edges. I say OK. And what I'm going to do is separate these variables into tiers where the only constraint I'm imposing is a variable in a later tier cannot cause a variable in an earlier tier. Anything else is allowable. And so I'm going to hypothesize that your socioeconomic status, this is of your parents, right, cannot be caused by anything else in this study, and your gender, or your sex, cannot be caused by anything else. So what other constraints might I entail? What other pieces of background knowledge might I put in here by maneuvering these tiers? What's your sense of it? And these are college plans. So I could put that in the bottom. Something like that. Now, do I know that parental encouragement can't be caused by college plans of the student? No, I don't know that. So by putting this set of tiers up, I'm saying that whatever the relationships that might exist, IQ cannot cause socioeconomic status. These are high school seniors or juniors. So the high school juniors' IQ cannot have an influence over their sex. I'm totally happy with that assumption. And I'm also happy saying that the child's IQ cannot cause their family's socioeconomic status. The socioeconomic status might cause the IQ because higher socioeconomic families tend to do more stimulation and practice tests, etc., but it can't go the other way. Parental encouragement can cause the sex of the child, but sex can cause parental encouragement. Especially in 1979, parents were more likely to encourage male children to go to college than they were to go encouraging female. Okay? So I think this is a reasonable set of constraints on the structures. So far, so good? So if I say done, now I put a search node in, and I say, okay, put the search and have input from the data and from the knowledge. So this knowledge I've just entered will constrain the search so that it will not entertain any of the structures that violate the background knowledge. And it'll search over all the structures that are consistent with the background knowledge. So if I say, let's allow latent common causes because there might be such, and I pick the FCI algorithm just for ease, and this allows me to set the alpha value, which we were talking about before, which is the way I do the statistical tests. 0.01 is a reasonable value to start out with. And then the number of bootstraps is set at 0 because I'm not doing bootstrapping yet. If I say that and run search and generate graph, this is what I get.
So, sorry, can you step back?
Sure. Okay. Let me copy the layout here. Go back. Say done. Okay, so you saw the data loaded, right? That's the data. And that was measured. And then the knowledge is going to constrain the search algorithm in terms of the structures it entertains as even possible. And so this says that socioeconomic status and sex cannot be caused by anything else that I've measured. IQ can't cause socioeconomic status or sex, parental encouragement can't cause socioeconomic or sex, and college plans can't cause IQ, parental encouragement, or socioeconomic and sex. Anything else is possible.
Is this direct and indirect?
Yes, direct and indirect. And so it doesn't mean that socioeconomic status might cause sex or vice versa. I've allowed that. I could forbid that within the tier by clicking on this. Now what I've discovered in the data is that they're independent, and so I've determined just by the data that sex can't cause socioeconomic status and vice versa. But I could forbid it anyway.
But that would be redundant in this instance, right?
Correct. So everybody clear on what the background knowledge is? And I say done. Now if I execute the search again, so this search is searching among the causal structures that include latent confounding, but don't allow later variables that I've specified as influencing earlier variables.
Can you redo how you set up the search? No.
Sure. So destroy the model. So now I have an empty search box and I have input from data and from knowledge. And I double click on the search box. And on the left it says show all the algorithms. And this is all of them. There's too many. So I'm going to allow latent common causes. And that brings me down to just 5. And the FCI is one of the simple that I'll default to. And then I have to set the parameters for that search. And in the first case, I choose an alpha value of 0.01, and that's to do the statistical tests. The maximum size of the conditioning set, I'm going to leave that -1 because that means any conditioning set. Now, if you have a humongous set of variables, like with 10,000 variables, if you might want to say, if these are discrete data, I lose power on the statistical tests when I condition on a large number of variables so quickly that I'm going to restrain how many variables I allow to be conditioned on, right? And that's what I can do with this parameter. And the maximum length for a discriminating path I won't go into, and I'll show you the bootstrap in a minute. If I say run the search and generate the graph, that's what I get. Now, what does this mean? And what's that funny green thing look like? Well, you've seen these circles before. So the first thing you'll notice is that socioeconomic status and sex have no connection whatsoever, which is, of course, what it should be. Right? It looks like sex has no influence on IQ, right? As I have 3 daughters, I will attest that if it has an influence, women are smarter. It has some possibility for influence of socioeconomic status on IQ, but it might be confounded because that's what that circle means. Socioeconomic status might have an influence on whether the parents encourage the student to go to college. IQ might have an influence, or it might be confounded, and sex might have an influence. But what does that green thing mean? If I click on this I, which tells me information, and I go to the bottom, if an edge is green, that means there is no latent confounder. So that means that the influence of parental encouragement on college plans looks direct and unconfounded with respect to this data. And so on the basis of this study, which was done in the 1970s in Wisconsin, the government made actually quite an important intervention to try to make parents be more willing to encourage their daughters to go to college. And this had a visible effect on the attendance rate of women on going to college. So it's a happy story all around. Yeah?
So, what's the difference between the green arrow and the blue arrow between SES and CP?
Right, so the blue arrow says, it says there's a relationship, right, that looks like it's between SES and CP, but we can't tell it's unconfounded. And the reason that relationship is there is partly because of the restrictions we put on the time ordering. But what I want to also show you is the facility of the bootstrap, which is extremely valuable. So in the set the parameters of the FCI search, there's a row that says the number of bootstraps. And if you do 0, which is the default, it just looks at the sample, runs the algorithm, and then puts the output out there. But if I say, let's run 200 bootstraps, what it's doing is it's going to actually bootstrap 200 additional samples, run the algorithm on each one, and then take the output that's most common for each particular edge. And then if I roll over the edge, I can tell what percentage of the time it did that particular output and what percentage it did other outputs. So let me show you how that works. So if I say 200, and I say run the search and generate the graph, now it's going to take some time. You're going to have to run the algorithm 200 times. Here it goes, and if I say layout, paste, now I get this kind of output, and I have an edge from PE to CP, but if I roll over it, I see that, well, it looks for PE to CP as a directed edge 75% of the time, and then with a circle arrow 24.5% of the time. So it's not every time it actually finds an unconfounded relationship from parental encouragement to college plans. It only finds that about 75% of the time. So it might be the case that random number generator seeds or ordering of the variables changed when you executed it when I did, and that's possibly why you got a different answer. But that gives you, I think, a lot of information, right, that helps you get some confidence over when you see an output, how likely is it to be a sensitive thing to the sample, or how likely is it to be robust?
So I wanted to give one final example today, which is...
Go on to display mode.
Oh, I'm sorry. Which is having to do with cancer, and give some background. These are all quite brief about cancer, and the Cancer Genome Atlas data set from NIH talk about the problem, analysis problem, and then a particular algorithm. You've heard a little bit about it. And then a plan, and then we'll look at some data and do a brief analysis of this cancer data. So as we all know, there are many cases of cancer. This is some data from about 10 years ago, but millions of cases of cancer just in the U.S. Breast cancer is the most common cancer in women, prostate cancer in men, but this just emphasizes the extent of the problem, which I think we probably all appreciate. So there's much to learn about cancer, and much progress and advances to be made in terms of understanding mechanisms and developing therapies. And I think it's an exciting time right now, actually, because we do have some of the measurement technologies that are allowing us to look under the hood of what's going on with cancer than we used to be able to. This slide shows it basically emphasizing that genomic alterations are at the core of much of what is driving cancer, changes in the DNA sequence information. And the plot that you see is basically showing somatic genomic alterations for different cancers. So this is bladder cancer here. The second one is breast cancer. This is showing, these are mutations or copy number variations. So the mean number of so-called somatic genomic alterations is around 300 in breast cancer. So if you looked at some breast cancer tissue and you looked at the DNA sequence information across all the genes, you'd see about 300 of them have either a somatic mutation or they have copy number variation that's pretty significant. And for different cancers, you have different numbers of alterations of different types that we won't have time to go into. But overall cancers, you have hundreds of somatic genomic alterations. If you look at the consensus list of known drivers, though, and you took the intersection with all of these, with the consensus list, you only end up with like 3 or 5 genes that are known to be drivers. So the belief is that there's a number of other drivers that we don't know about yet, and understanding those is important because those are effectively the root causes of much of the cancer, much of the cause of cancer. So we're looking for these, basically these root causes. The cancer genome atlas is a comprehensive, is quite a big set of data that was collected by NIH through NIH support and sponsorship and coordination. And it's really trying to understand the molecular basis of cancer through measuring many different kinds of omics measurements. It was started about 12 years ago. It's basically completed, and the main goal is to try to really improve our ability to diagnose, treat, and prevent cancer by understanding cancer molecular mechanisms. So it was part of NCI and NHG RI's efforts through NIH. Here are some of the cancer types that have been collected in TCGA. So these are just in alphabetical order, but you can see breast cancer here, there's about over 1,000 cases of breast cancer. So this was a huge effort, and different institutions around the US were involved in collecting these tissues and in measuring different omic measurements. I'll show you in a minute. So you can see there's many different kinds of cancer and hundreds of thousands of cases. Overall, I believe there was around 10,000 cases that were collected. This data is actually available through NIH to anyone. You have to apply for it, but if you follow the procedures, you can get access to this data, and it's still quite valuable, and researchers are still studying it. So this shows some of the types of data. So there's whole exome data, and this breast cancer is 1,000 plus cases. SNP data, methylation data, RNA-seq data, microRNA data, and some clinical data as well. And so in terms of the analysis problem that I wanted to talk about, of course, the basic idea is that we've got damage in the DNA leading to expression changes. Those expression changes then drive the cells to behave in cancerous ways, leading to clinical cancer. So the somatic genomic alterations in the DNA are largely responsible for initiating cancer, and the SGAs cause cancer then through these differential expression of genes, or DEGs. And those SGAs that cause DEGs, often and strongly are good candidates as drivers for cancer. So this analysis problem we're going to look at is to find SGAs that cause DEGs in breast cancer under assumptions. And as I mentioned before, the genomic drivers are really can be viewed as the root causes of cell signaling pathway aberrations. Those pathways are controlling cell function, and when they're aberrant, they can cause cell dysfunction, including cancer. And the goal would be to understand these and then be able to use them to develop therapeutic strategies for targeting those pathways. And if we understood them in a given tumor, we'd be able to develop precision medicine kinds of ways of taking the medications that we have and targeting those pathways to try to intercept the cancer. This is, we'll look at just a simple bipartite graph where on the first level we have SGAs, and on the second level we have DEGs. And so what we're interested in is what are the relationships between the SGAs and the DEGs. We're actually going to look at the relationships among the DEGs as well, just for the purposes of illustration. So the way that these SGA variables were measured in the data set that you have is that, as I mentioned, a SGA is a non-synonymous somatic mutation, or it's a copy number alteration, and if that exists in a gene in a given tumor, it'll be given a 1, otherwise it's given a 0. And then the DEGs are defined as differentially expressed significantly over normal tissue for that gene. And basically we used a significant, I think it was 2 standard deviations above or below the mean for the normal tissue as a to code a 1, otherwise it's coded as a 0. And some of the main assumptions then are that the DEGs are not causing the SGAs, and that the SGAs and DEGs are not confounded. The greedy equivalent search is a method that was developed by Chickering and others, and it searches over equivalence classes of Bayesian network structures, patterns that we talked about before, using a Bayesian score. It's a greedy algorithm with a forward-stepping phase and a backward-stepping phase, and it has some guarantees under assumptions of finding the data-generating causal model. The FGES is an optimized version of GES that's optimized in different ways on a single processor, but also is parallelized to run on multiple processors. So we're going to be running FGES in our example. And in terms of the data set you have that's called the cancer data set, the way that we created that is that we just basically took the breast cancer cases from TCGA from these data sources, and we selected those tumors that had measurements for whole exome, copy number, and gene expression. And out of the 1,000 plus, we ended up with 851 tumors. And then the variables for the SGAs I've basically already mentioned. In terms of copy number variation, there's a GASTIC score that was used to determine whether or not there was a significant copy number variation. But we just selected, for the purposes of illustration, to make it easier to look at the graphs, we just selected 6 SGAs that are well known to be breast cancer drivers. And for the DEGs, we basically used a program that we've developed called TCI that finds associations between SGAs and DEGs. And we basically kind of pre-filtered to find DEGs that were oftentimes associated with SGAs according to that program. So there's been some filtering done to get the thousands of DEGs down to just 60. Again, for the purposes of us being able to kind of look at it and illustrate it in this exercise. So the analysis plan then is for you all to load the data, and I'll go through it myself here and show you the steps, add some background knowledge, and then apply FGS to the data set, given that background knowledge, and view the causal network that we obtain. Okay, so now let's look at some of this TCGA cancer data that we've simplified, and you should have a file that we made available to you called cancer_data. And what we're going to do first is load that data. So click the data box here, bring it over, and then click it on the left click it on the screen here, and then double click on this. And you'll see a spreadsheet. Well, we don't want to enter it manually, but we're going to go up to File and say Load Data. When we do that, we see cancer data here. We're going to click that, and your file structure may look a little different, but we're going to choose that. And we get a bunch of options for the data loading. It is tabular data. It's not continuous, it's discrete data, so we're going to click that. It's tab delimited, and all the others should be okay. So we're going to ask it to see if it validates, and it does some tests, and it does. It passed with no error. So we're going to say load, and what it does is it loads that data from the file into this matrix. And you can see that we have some SGA data that we talked about before, the 6 SGAs that we had selected, and then we have a bunch of DEGs in the other columns, and we have the rows, one for each sample. So we're going to say done, and now we've got the data loaded. We also want to have some knowledge, and so we go down to the knowledge box here. This is going to allow us to make a bipartite graph, a 2-level graph. And so let's click that here, and we're going to come over and left click on this arrow and hold it down and then drag it over and release it. And what that's basically going to do is let the knowledge be informed by the data, which is really the variables, the variable names. So when we go into knowledge, we're going to use tiers and edges. I'll explain that in a minute. That's the kind of knowledge we're going to use. We get this dialog box here, and you can see above it basically brought these variable names from the data set. So that's what that arrow was doing, was informing the knowledge about what we're going to have knowledge about. If you left click one of these and you hit Control A, it will select all of them. Now admittedly what I'm about to do is a little kludgy, and we're in the process of making this more efficient, but right now this is the way you need to do it in this version. So you drag that, left drag and drop all of those variables here. And what we're going to do is we're going to put those in tier 2. And in tier 1, we're going to drag and drop, left drag and drop all the SGAs. Because we want those to be in tier 1. Here's another one. We've got 6, so we need to find all 6 of them. Here's another one, pick 3CA. Here's another one, RB1. And we've got one more in here. Here we go, TP53, a very important one. So here we've got 6 SGAs, and then we know all the rest of them are DEGs. Putting them in tier 1 and in tier 2 basically says that it doesn't allow the program, when it's searching, to have something in tier 2 be a cause of tier 1. But something in tier 1 may possibly, it's allowed to be a cause of something in tier 2. And that's what we want. We think if there's causality, it would go in that direction. We're going to forbid arcs within this tier. That means that we're not going to try to learn any relationships among these SGAs. We're not, right now, interested in that. And we're going to allow edges, causal arcs, between the DEGs and between the SGAs and the DEGs. So at this point, we're done with the knowledge. We can click Done here. Now, we'd like to actually do some search for a graph. So we click on Search, and we bring it over into the workspace and click here. Again, get the arcs. The search is going to be informed by both the data and by the knowledge. So we put arrows into search from there, and we double click on search. It gives us a bunch of algorithms that we can choose from, and here we can sort of select features that will create a smaller list here. So for example, rather than show all, we might say forbid latent common causes, allow latent common causes, and so forth. So we're not going to try to model latent common causes right now because we think that the way this problem is set up, it's unlikely that there's confounding of the SGAs and the DEGs. That could be arguable, but we're going to assume that that's not going on. And we're going to use FGS, which we've mentioned before. Click that, and we're going to say Done. Okay, so it's telling us we haven't done a search yet, so we just selected it. So we need to come down here and actually set the parameters. And there's a number of parameters. We're just going to assume the defaults for the parameters right now. And the manual goes into those in more detail, and you can look those up. But in the interest of time, we're not going to go through the parameters. And we're going to say Run Search and generate a graph. So let's get out of this and go back, and we're going to generate, using the graph operator, we're going to generate a little bit more sophisticated display. So we'll bring a flow arc into graph from the search results. We'll click on this, and we'll look for a graph. And here we get a graph for which we have some more options. So for example, we can go up here in terms of layout and try different types of layout. Here's causal order, and that gives you sort of the root nodes, and then the nodes that follow from that are shown. And another one that, so I'll then we will look at, let's look at another option here. So if you look at distance from selected, for example, if we save GATA3 and we look at distance from the selected node, what we will get is that node up at the top, and then basically the different levels that it affects. And in that sense, you can, by doing that, you can get a sense of sort of how far away it is in terms of causal causal influence or for some of these other nodes. That can be useful if you have a particular node that's of interest. So you can try these different layouts and see which ones look to be the most informative for what you're interested in studying. So we'll say done on that now. So at this point, what we've been able to do is take some data and some background knowledge, do a search to generate a graphical structure, and then be able to display that structure in various ways. Some of the other things that we could do, but I'm not going to demonstrate for in the interest of time, is we could click on the parametric model and take the graph, the structure that we learned, and have that be an input, and the data, and tell what parametric form we want to parameterize that structure with, given the data. And then we could draw an arc from that parametric specification into something that's called the estimator. And basically what that will do is we draw an arc from the parametric model and from the data, and that estimator will basically estimate the distribution in the data. So this, I hope, gives you an idea of how you can take, in this case, some real cancer data and analyze it for some relationships that you can then explore. Of course, with real data, there would be many more variables and more complicated graphs in this case. But I think we've demonstrated some of the basic capabilities that you typically use to explore this kind of data. So thank you. And one other thing, if you see this, this is on the slides, but at the bottom is a URL where you can get plug-in for Cytoscape. Some of you may use Cytoscape. It's a very popular program among folks in bioinformatics. Cytoscape has some very nice layout programs. I mean, Tetrad has some pretty good ones, but Cytoscape's got some great layout programs. And so there's a plug-in for, you can export one of the network graphs to a file as a JSON file and then import it into Cytoscape, and you can use all the functionality of Cytoscape to view the graph. And this shows in our problem, this was under some different settings of the algorithms and running the algorithms, but you can see here TP53 and all the different DEGs that it's driving under that algorithm run, some of the other SGAs here and how they intersect in causing some of the same DEGs and so forth. So Cytoscape that's a really nice way to view your graphs. So at this point, we really wanted to just sort of summarize the workshop and give you some pointers for additional information. So I think Richard's just going to get started. Okay.
Finish up.
So at a high level, what we've done is, or hope to do, is giving you some information about how you represent causal systems using causal graphs, how they relate to allowing you to represent observational relationships as well as relationships under interventions. And we talked about a little bit more specifically causal Bayesian networks as one type of causal graph. Richard talked about the causal Markov's axiom and just touched on deseparation, but if you were able to do the online exercises, you learned a lot more about deseparation. And for any of you interested in this area, I would highly encourage you to do that online tutorial, because it really gives you a good sense for what the representation is and how to look at a graph, a causal Bayesian network, and read off from the graph, what it's telling you about the relationships among the variables. We also talked a bit about search, how to generate causal structure hypotheses using search through these algorithms, and determine the causal structures that are consistent with testable constraints in the data. In terms of additional pointers, let me talk about first some with training. If you go to the CCD website shown here, you'll see that there's some additional tutorials. And actually, Richard gives a summer week-long short course at CMU. And this has the complete short course from 2016. So if you want to get additional information that goes into more depth from what we've covered today, that's a good a set of videos. And then there's tools. So Tetrad is the most recent version of Tetrad is located here at these tools. But there's also other tools for using like Tetrad algorithms in Python and in R. And also there's ways of using it in a command line fashion, so that as a Java command line, so that you can basically incorporate these algorithms into your programming workflow. You don't have to use a desktop interface. Suggested readings, there's a lot of possibilities, but I wanted to sort of boil it down to some that I thought would be the most useful next steps. The first one by Lagani is a nice paper from a couple of years ago, and it's called Probabilistic Computational Causal Discovery for Systems Biology. And they give 3 very illustrative biomedical examples of how to use these methods. They also go into more detail about what's going on in terms of the assumptions and what's going on in terms of how the algorithms work, both Bayesian and constraint-based. So I think it's a nice next step after this tutorial. The other one is a book by Pearl and others on causal inference and statistics. That's a nice book on the representation itself, talking about causal models, causal Bayesian networks, what they can represent, how you can use them for inference. And it's a short version of his longer book. And I think it's a fairly accessible and easy to read and get through book that talks about modeling, causal modeling. There's a number of conferences that go into report research in this area, the uncertainty in artificial intelligence, AI and statistics, NIPS also. NIPS and UAI oftentimes have causal modeling work and discovery workshops associated with them that occur right before or right after the main conference. And then there's a workshop that's held in Europe on probabilistic graphical models that's quite relevant. So I just wanted to really finally acknowledge the NIH that supported much of this work through a U54, and also acknowledge Dr. Chun-Hei Kai in the CCD for his help with the cancer example and getting that all pulled together. So thank you very much for attending, and we can take questions.